So let me just see. And the effort is a global one of cancer genomics. When it comes to the oncogenic signatures, OSCs, just a label to remember in a little while. But this is a global effort, and in the US, where I currently work, it's called the Cancer Genome Atlas. And it goes all the way from a collection of tissues. There's a whole set of laboratories, 15. It's like a blueprint network, like a framework program, whatever the current number is, 9, a program. Many labs are participating. They get funding. And it's a very interesting collaborative exercise, actually. It actually works and is productive. It is coming to an end, though, in the middle of next year, but the international effort, the ICGC, International Cancer Genome Consortium, is going to continue beyond that, and as you see in a minute. So you go from tumor samples, which are basically surgical samples, primary surgical samples from real patients who have cancer. Most of them in this effort are primary, not metastatic. It's the metastatic that kills people, but these are primary just to see what happens initially in tumors. And so each one of these samples represents a person who had cancer. In some cases, the cancer history is known. And the question is, what's going on at the molecular level, making use of all the technologies that we now have, going beyond what was originally known. Of course, lots of things have been known about cancer, but this is now looking at molecular detail and then making the link back to the system that is the organism. And so let's look at some of the molecular detail. It's done by sequencing primarily and array technology initially, now mostly sequencing. Different groups are participating, SNP arrays in Boston. People are doing RNA-seq, of course. People are doing DNA methylation. This is Peter Laird and colleagues, you know him very well. And microRNA sequencing, as well as low-pass whole genome sequencing, get a copper numeration, typically paired in low-pass, Raju Kuchalapadi and others. This is reverse phase protein array, which is an antibody-based. It's a cell lysate on chips. Each chip is interrogated with an antibody. So it's protein and phosphoprotein readout, limited, however, to about 200 proteins. But it's beginning. Of course, mass spec proteomics would be the right way to do it, but that's more expensive, but that's rapidly developing, as you know. And then genome data analysis centers, people who do computational biology and systems biology distributed around the US that are participating in this effort. And then, of course, this is in part embedded in the ICGC, organized by Tom Hudson out of Toronto, and it's a fantastic international effort. I love this because the whole planet is involved in this, and it's moving in the direction of tens of thousands of tumor samples. And of course, technology is going to improve, as is the fullness of completeness of data. And there's a major missing piece, I might as well see it now, which is in terms of epigenetics, it's only DNA methylation in this large project has been measured, no histone modifications. And no other covalent modifications, actually, that might also be of interest. So this is the current spectrum here over the last 5 years, ending in 2014 in the US. The different tumor types, each one about 500 individuals. This is going to approach 20,000 tumors fairly soon. And so let's look at what we can learn from the molecular profiles of each of these tumor samples. So here is just to start with one particular data type, which I picked for the event, which is Peter Laird using an Infinium 450 platform. These are the individual features on that DNA methylation array and method, and these are the individual samples clustered according to the similarity of these columns, and red is up and blue is down. And so in terms of DNA methylation, and these are 176, in this case, prostate cancer, prostate adenocarcinoma, and 49 normals. You can see immediately in terms of the DNA methylation pattern that there's a patch here that looks like the normals, actually. That's interesting. We'll come to that in a second. So these are actually, now we checked with pathologists in great detail, are these really tumors or not? And they are tumors. So these are early ones perhaps, are certainly less aggressive, as you see in a moment. And the other patterns here, you see there's a cluster here that comes out. It's a fairly good cluster. What is going on here? This is DNA methylation, so how does it relate to anything? Let's turn this around. Now we're having the samples, the patients going vertically, and these are the features on the Infinium array, a subset of those. And compared to copy number changes, this is now the human genome left to right. For each of these samples, each row is a patient, you see amplification of DNA, pieces, DNA fragments, copy number changes in red, and deletion, mostly homozygous deletion on this graph in blue. So you see that actually there is a relationship between DNA methylation and copy number changes. The most striking one is this one here, that these guys that look like normals, in fact, also in terms of copy number changes are diploid. They look like normals. And we've shown a few years ago in the paper, Barry Taylor and Nikki Schultz et al., that actually if you order prostate cancer sample by the degree of copy number changes in the genome, just as a linear scale, there is about 20 to 25% of samples which are very quiet in terms of copy number change. We see here the relation to DNA methylation. And those patients, even though actually they had prostate cancer and they had surgery, this is not made up, another medical mistake, those 20, 25% have excellent prognosis. This argues in terms of the treatment of prostate cancer that if you can turn this into a reliable test, and I think it can be, even though this has been done, you could actually not do surgery for about 1 in 5 men who currently get prostate cancer surgery. But it's not on Gleason grade, data not shown. It is actually based on the molecular profile, which is much more reliable than the pathology, actually. And so this is an example of where a very simple assessment can give you some insight into clinical outcomes and possible different choice of treatment. Then there's this other group here, and you see that those DNA methylation patterns actually relate very much to a much more unruly pattern of DNA copy number changes. Details difficult to unravel. We don't understand it. There's a group here that has some characteristic deletions here in chromosome 2, 5, and 6. We haven't unraveled that in terms of the gene correlation to either the mechanism of copy number changes or to the patterns of DNA methylation. These have worse prognosis. So that's just a quick survey. There's lots and lots more about DNA methylation that we could tell you about. Peter Laird has written very nice papers on that. Others have, Steve Balin and so on, and others. I think, however, that, again, I'll come back to that very briefly at the end, I'll say it now. I think that epigenetics is just under-resourced in cancer genomics. I think it would be good to have more collaborative links between the cancer genomics community and the epigenetics community. Many of the things that I've learned today and I've heard are actually underused, is a mild term, in cancer genomics. So let me give you one story of how one now can look across all these different tumor samples, 3,500 and rising. We just limited ourselves to a kind of a frozen data set, 15 different groups participating, 35 papers submitted, 17 were published. People, this is a liberation of effort rather than just having these marker papers like in ENCODE. You have a marker paper and then you have a basket of papers co-published with it. And everybody is enthusiastic, and the postdocs get their, the micro-coded author positions and so on. And so it helps actually motivate people to have these baskets of papers in these large efforts, such as yours. And I don't know how you organize it. And so in this one here, what we try to do is take the description that we have currently. Some aspects are missing, but these are the main data types that are the richest one. Copy number alterations, somatic mutations, which are amino acid changing mutations for the most part, or truncating or splice site but not silent mutations, and DNA methylation by the subset demethylation using the technology that I showed you, and then across 3,000 different samples. So again, each one of these vertically now is actually a tumor sample, is a patient sample, and these are the different tissue tumor types. Glioblastoma, colorectal, breast cancer, bladder cancer, kidney cancer, lung, 3 types of lung cancer, ovarian, and endometrial. And so this is rich data. This is actually about a billion data points. Actually, the original data is even much larger because all the sequencing traces. So this is massive data in some sense. But of course, what you want to do is analyze this in a way that makes sense. And what we try to do is actually to simplify this analysis by bringing in prior knowledge of what we know about cancer biology and a bit of statistics. So this massive data, in this case, becomes much more transparent if you simplify the analysis by looking at a subset of the data points. And we do that by using our background knowledge of what we know about oncogenes and tumor repressors, but also statistics. And simply, you count those things which tend to be recurrent. So if you see something in one of these vertical columns, some event here that is present in many of the columns, say this one here, or down here, not visible, something that's recurrent across many different tumor samples. This can be independent of tissue type, you take those more seriously. So you take this massive data set and you reduce it down to a description in terms of what turns out to be about 500 selected functional events, potential functional events. And these events are where a gene is mutated somatically, or the copy number altered, large high amplification, homozygous deletion, or it has where we've seen DNA methylation that actually anti-correlates with mRNA levels in a very clear-cut way. So a conservative subset of events which you lose information, but you gain a lot of signal to noise because you're looking at things that are plausibly functional. And now let's look at those thousands of tumor samples across different tissue types and see whether we can make sense by simply clustering and using a clustering algorithm that actually addresses the computational difficulty. So here you have these 3,300 tumor samples. You have each one described by 500 numbers. And now you try to group them into tumor samples that have interesting collections of alterations that might be therapeutically actionable. And the algorithm is slightly non-trivial because when you reduce things in this way, it's difficult to use standard sort of Euclidean matrix. So Giovanni Cerielli invented an algorithm, it's not the only one, that basically it's a bipartite graph, you take alterations and samples and simply try to connect alterations and samples in such a way that you get groups, subgroups, clusters, such that there are many connections within within the subgroup and few connections in between. So simply, basically, you're trying to come up in the end with sets of alterations and set of tumor samples that are highly connected. That is, one is associated with the other, and with as few cross connections as possible. It's like cleaning up a matrix, making it block diagonal, or a rectangle, making it block diagonal. This is the algorithm that he used. It comes actually from the social sciences, and it was adapted. And then you get a hierarchical. So this is a different way of clustering than you typically see. And also the other difference is, rather than just clustering everything into the optimum number of clusters, 3 or 4 or 5, what you do a hierarchical decomposition that gives you some insight. So first, let's take the decomposition of these 3,000 tumor samples using these 500 events into 2 subclasses that might be interestingly different, and then further down the tree. And then look at each of these levels and see whether you see something interesting that might relate to potential therapy. So the first observation is you get 2 main subclasses, this one here and this one, we call it the N class and the C class. Why? Because if you look at the events in terms of the statistical significance, either in one direction or the other, I mean, either for one or the other, in this cluster, if you collect them, you see that most of them in this one class, in the top class, are, and the green color here means mutations, and most of them in this other class are, and the red and the blue mean copy number changes. This was quite surprising to us, and even though it's not uniformly high, there's a tendency that's very striking. And so, if you generalize that, certainly at the extremes of genomic stability, in this plot here, each of these squares is sets of samples, and the samples are characterized by how many mutations are in the sample that are somatic, that change the protein sequence, and how many copy number changes that are amplifications or deletions. And so each tumor typically has, this level of mutation, this level of copy number changes, and so you can organize them that way. Well, this upper right is completely empty. You have no tumor samples where you have a very large number of mutations and a very large number of copy number alterations. And that was a surprise. And so it looks like there's a magnet. One interpretation is that at the extremes of genomic instability, when the number of mutations are large, this is a logarithmic scale actually, so there's quite a factor here, or the number of copy numbers are very large, you have one process that dominates, and those cancer cells just run away. At the lower level, there's some kind of a mixture, and you get a preferential tendency to have either copy number changes, DNA fragments, genome being rearranged, or you have mutations that affect the properties of proteins in the cell. And so now, let's look in more detail. This is a very, very complicated slide, and since I apologize, but what you see here now, these 2 main classes, and then broken down into 7 subclasses, which are these rectangles, then 31 subclasses beyond that. Now, for each of these subclasses, you can go along. Now you can ask yourself, say, for this set of tumor samples, which is maybe 30 or 40, first of all, what tissue types are involved? And typically, there's more than one tissue type. Secondly, what are the genomic alterations that are involved? Here are the mutations, and here are the copy number changes. And say you take this one, M7, whatever it is, and you look up what are typically the mutations that arise in here, or down here, another class, what are typically the copy numbers. It's not black and white. If it were ideal, you'd have a diagonal form of this matrix here, have a certain small set of mutations relate to a set of patients. It's not black and white, but there's a very strong tendency here. So there's certain mechanisms of oncogenesis, plausibly, that you can read off here by looking at groups of alterations here or here that relate to a group of patients, and those patients typically are not, this is not a diagonal in terms of tumor types against subclasses, but this is mixed. It's quite strongly mixed. So you can have, for example, endometrial cancer, polymerase epsilon mutated. You can have colorectal polymerase epsilon mutated. It's a subset of both of those tumor types. They both have a very high mutation count, indicative of a mutational rate in oncogenesis. And they have alterations that are characteristic as a group. And so you want to treat patients from those two tumor types potentially with the same therapy. And there are quite a number of examples of that. And the examples are written here on the right. You see there's an attempt to actually associate this set of characteristic alterations that are characteristic in these rows with a group of patients, relate that to pathways, which I could tell about at length. These are the well-known cancer pathways. In fact, one of them just came out in prostate cancer. It's a SWIFT having to do with chromatin organization that I suspect you're familiar with. It turns out there was a paper last week. It has a very interesting signature in prostate cancer, etc. Many fascinating examples. When you look in detail, you see that, well, this is the chromatin organization, and this one is one of the very, very common cancer alterations. You see that actually there's a very small number of genes that dominate in this subgroup of about, of 6 subclasses that grouped together perhaps might enter clinical trial, and you would have to collect patients from different subgroups. Perhaps most of them here in this here would be endometrial cancer, but you also have head and neck and one other. This might be, because it's head and neck and endometrial, might be virus associated. We haven't looked that up in particular. But you see that in each of these rows, each of these subclasses, you'd have to take patients from different tumor types. And that's a new kind of clinical trial. I'll come to that in a minute. So you go on, and you have very interesting patterns. I mean, the details are, from my point of view, fascinating in terms of the biology. This is the polymerase epsilon, which is a particular mutation. We actually co-discovered that in this process with a group who founded by a GWAS study. And MLH1, the MLH1 group is very clear as a subgroup. ARID1A, you're familiar with that one too. CTCF, in fact, co-occurs with mutations in the PR3 kinase path, PIK3CA and PIK3R1, in this particular subgroup, M5, which shares between endometrial cancer. The CTCF, yes, these are subgroups of endometrial cancer. And so each of these as a row would indicate a common oncogenic mechanisms, potentially a similar response to therapy if you have an agent. And similarly for the copy number changes. Let me just go on in the interest of time. Interestingly, in all of the tumor samples, all these subclasses that have very highly copy number rearranged genomes, there is an exceptional mutation. P53 is always mutated. It is a very strong confirmation that there's a link between genomic instability at the level of copy number fragmentation and P53 mutations, with P53 plausibly being an early event. High-grade serous ovarian cancer is a prime example of that. So jumping forward, you could now take one of these classes or two of these classes, whatever the right unification is, look at the main alterations and come from looking at the previous graph and now look for drugs that might be available. Say you may want to treat these patients here in head and neck and breast cancer with a combination of PI3 kinase inhibitor and mTOR inhibitor, etc. Those in the field of cancer therapy recognize that some of these combinations are being tried. So this is not at all completely new territory. But the suggestion is twofold. One of them, treat people systematically across different tumor types with similar agents. And number two, do this computationally using the fantastic wealth of information that we get from the cancer genome atlas and ICGs. You do it systematically and aggregate, but perhaps look like a minority position of just a few samples in this subtype, a few in this, by few I mean 1 to 5%, aggregate them laterally, and then create clinical trials that then have fairly large numbers and are meaningful, where typically pharma, big pharma, would certainly not go there because they would just have a small number of patients. But some of these are highly effective. And I could tell you stories about that are anecdotal, that are as interesting as the RAF inhibitor in advanced melanoma that works as a single agent in patients that have a BRAF V600E mutation, but that leads to recurrence, hence a strong argument for combination therapy, which is what we're working on here and also in the wet laboratory that I run. And so coming to the end, so just again to make the point about how this now leads to a potentially clinical trials, which are departure from previous mode of clinical trials. Typically, this is just a cut out of this pan-cancer analysis, where in these squares it shows you the number of patients in luminal breast cancer. 18% have a mutation in FGF1R or an event in alteration in FGF1R, or 80%, not surprisingly, in the HER2 plus subtype of breast cancer having mutation in alteration in ERB2. This is what is already known, now more comprehensively, but this is the typical organization, tissue types and alterations, and then perhaps now let's change that and now go from the tissue types to these classes, so we can now use the advantage of aggregating across different tissue types, same kind of alterations, green are the mutations, red are the copy number changes here, mostly activations, and if one, the tumor repressor would be deleted. And the idea would be to take one of the, say if you had a drug against KRAS, which you don't, you would take patients and aggregate them across here. If you have a drug against FGFR1, which I think there is one now in progress, you would take patients from these subgroups and no longer rely on tissue characterization necessarily. And so we're going from what used to be the picture with the different tissue types and different alterations, it's a more complete picture, to one we're suggesting that these derived classes might be more effective in terms of inferring what the oncogenic events are, number one. Number two, picking single agents and then combinations of agents that are effective, where the combination is aimed at blocking the resistance to therapy, which typically arises in the otherwise wonderful anti-cancer drugs such as the RAF inhibitor or the AR inhibitor. Okay, that's it. This is acknowledgements to people who did the oncogenic signature analysis. Let me just show you a useful tool just 2 more minutes, a useful tool you might want to use, cbioportal.org. It's an information system that pulls together all this information, but in a way that's consumable by people who are doing research at their desktop rather than having to write programs or do massive analyses or stare at clustergrams. It's a very convenient way of looking at their genes here and how they're altered across different tumor samples or individual tumors in the vertical direction. It gives you tools to analyze mutations in the context of sequence alignments, 3D structures, survival plots, et cetera, DNA relation. Also, here's an example, CTCF, one of your friends. This is just, I did this afternoon actually. This took about 5 minutes to make the next 3 slides. You get the distribution in these different tumor types of mutations or homozygous deletion of CTCF. And much of this is not understood, I'm pretty sure. I looked at the mutations here, again, just today. Here are the mutations in the zinc. This is across all tumor types, thousands of samples. This is now more than 10,000 samples. The 9 or 11 zinc fingers, or whatever they are, around 10 zinc fingers of CTCF. You can see some recurrent mutations. This is truncating in this particular zinc finger. In this zinc finger here, there's a mutation that occurs quite frequently, possibly affecting the DNA specificity. You can use this tool, do this analysis yourself. You can learn it in a short time, and it's a very convenient way of getting to the kind of information that otherwise would take a long time to pull out. And it's just evolving in the direction of having patient reports, where for the individual patient you see copy number changes, mutations, and details. And if these details are understood and well prioritized, you can pick a combination of drugs, ideally, that would address this particular patient's. But now we have the data, we can actually do this and not just talk about it. Okay, thanks very much. And collaborations, again, not for myself, but speaking for the cancer research community, knowing a bit of what happens in TCGA, epigenetics has to come in a much more erudite and active way. Thanks.
Chris, thanks a lot for this wonderful overview of this very complex world of cancer biology.
This is a great talk, thanks. I have a question about the cancer signature classes and subclasses. Do they tell you anything about when you have escape from monotherapy, what that pathway is going to be? Does escape move you to a different class or subclass, or does it hit different things in the same class and subclass?
So there are examples of both, but there are also the clear examples, and your question indicates that where actually another oncogenic mechanism kicks in. Just a friend of mine, just ex-postdoctoral manufacturing PhD student, has just, this is not the only example, it's someone I know well, a low-grade glioma, and taken from one individual, take several samples, and you look in disease progression, and you see things change actually, and you see things switch from one program to another. I have a slide also that shows that in prostate cancer. I could show you offline. It was actually in the deck. So a bit of both. Sometimes it's just the same pathway being activated by another mechanism if you block a particular path, but often you have an escape that goes to a parallel track, which is sometimes quite complicated.
I have a question. The SIMP methylator phenotype, does it contribute more to the instability segment? So is it a very informative and very important parameter to classify for you between the C and the M's?
Is it important for the classification? Yeah. Well, in terms of oncogenic mechanisms, it's a revelation in a way. But often we don't know the precise mechanism. We know it in some cases. MLH1 or POLE, those are two examples where we know what the mechanism appears to be of genomic instability in terms of POLE, its copy number, it's mutated. Well, MLH1 is as well. So these are defects in error correction during DNA replication. So in those cases, we know the mechanism. For example, in terms of copy number changes, high-grade serous ovarian cancer and basal or triple-negative breast cancer share amazing similarity in terms of just copy number changes being all messed up. The DNA fragment. We don't know the mechanism, even though there's lots of speculation about BRCA1, BRCA2, P53, but the precise details aren't known. Maybe I'm not answering your question, but...
No, I was a little bit pointing towards the changes in the epigenomic landscape. So DNA methylation, particularly CpG island de novo methylation is observed with SIMP positive, SIMP negative. So instability or non-instability. And I was wondering whether this is also a major discriminator for you.
So yes, and I think it's incompletely understood. One example you may know is IDH1, IDH2, IDH1 mutation, first occurs in glioblastoma and also in AML. There's a complicated but pretty obvious now linkage between a mutation in this enzyme, alpha-ketoglutarate, and then affecting a DNA modification. And so I think there are links to be discovered between what might often be the original alteration in terms of copy, in terms of genes being amplified, deleted, or mutated, and then effects on the epigenetic landscape. Some of them are absolutely striking, like the example I showed you in prostate cancer, where there's a clear distinction. But there are lots of details there that I think have to be worked out.
Okay. So if there is no further question, I would like to close the session. Thank you again for your presentation.